Ready? Steady, go. Okay. We began talking about a lot of the different dimensions in V1. Let's just sort of summarize and put them together so that we got them. We have retinal position, eye of origin, ocular dominance columns, orientation columns, complexity, going from simple to complex to who knows what else, depth perception, color perception in the blobs. How can we map it all in a single 2D sheet is the question. Okay? And the big answer is jitter. Okay. So.
Is the cortex 2D or?
You're right. It's 3D because it has depth. So it's 3D. Right. Okay. Why do we call it 2D? Because you're right. It has depth and it does have a third dimension, but it is very thin. Okay. But yeah, you're right. It does use the depth. Okay. Good. 3D. Just that we shouldn't mix up. It's 2D over position in the world, in the half world, and it's not 3D in terms of depth. The depth of the cortex has nothing to do with depth in the world. Okay? So here are the ocular dominance columns, stained by putting stain in one of the eyes and having it transfer from the eye to the LGN to the cortex, and therefore it can be stained. And we discussed what happens if you take a kitten, blind it in one eye, and the columns shrink and expand. Okay. Next we had the, why am I here? I'm supposed to be. We have orientation selectivity in orientation maps, and the nice work done by Amir and Greenwald showing that you have kind of pinwheels where you have all the different orientations around one point, trying to associate this point with the color blobs. Probably not true. Okay. But there are these areas where we have color sensitivity and not orientation sensitivity, and around it the orientation columns. And there is a kind of orthogonal. This is trying to show that this area is left eye, this is right eye, this is left eye, and these areas, left and right eye, are orthogonal to the differences of the orientation which are within each one of them. Is that clear? No. What I'm saying is we have left eye, right eye, left eye. Within the left eye area, we have the different orientations being represented. Within the right eye, we have the different orientations being selected. Okay? Do you see here? Sort of. So the typical picture is we have a left eye and a right eye. Right and left. And we have different orientations. This is vertical, oblique, horizontal, vertical. Okay? Now this is as if it's exactly in a square, what I call the ice cube model. Okay? But it's not square, it's contorted. The important thing is that within one such area of cortex called a hypercolumn, we have both ocular dominances and all the orientations. Okay? So now the question is as follows. If we have this hypercolumn, which has both left and right ocular dominance and all the orientations, and this is part of a map of the world, which which is seeing from the midline to the periphery. Okay? We know that we map the whole world over the cortex. If that's the case, then we can expect that we can only see with the left eye vertical things in this point of the world, and if we move to the next point of the world, we'll only see with the right eye vertical. And if we move to another point of the world, we'll only see with the right eye horizontal stuff. So how do we map both left and right and orientation and a map of the world? Got the question? Am I losing you? No. Okay? What's the answer? Jitter. The answer is jitter. That this map, these maps are not well, let's talk about the position in the world map is not precise. When we go from position to position in the cortex, we do not go from position to position in the world precisely. Okay? So that if the receptor field size is something like this, then we can expect that the I can't draw both at the same time, both the position in the cortex and the position in the world. Okay? So if this is the position in the cortex, then this cell will see that position, and the one next to it will not see precisely the one next, and the next one precisely. But there's a jitter going around of the position. As I move across the cortex, the position in the world is not precise but jittering around. And therefore, this area of hypercolumn will cover all of this area of the world in all the directions. Is that clear? Okay. But by and large, when we go from hypercolumn to hypercolumn, then we have also moved to a new position in the world. From cell to cell, we have not necessarily moved from position to position. But from this cell to this cell, we have. Okay? So there's overlap between the positions in the world. Okay. Sorry, but that's, yes. I didn't say that. I said that the cells are not aligned one next to each other according to the precise position in the world. Okay? Maybe their connections are. Okay? So the cells might know. Do we know? Another question. Okay? Okay. And depth in the cortex. As you said, there's a third dimension. The depth in the cortex has to do with the complexity, going from simple cells to complex cells. Okay? From input to output layers and different kinds of processing that take place. You know, the input layers are the area layer 4, and the output are from higher and lower, and different processing taking place there. And as I said, we still don't know everything about it. Okay. One more thing. A few more things. Okay. First of all, Yuval and Wiesel spoke about simple, complex, and then they said there's hypercomplex, more complex than complex. And what they were trying to say is that the whole of visual processing is a matter of going up in complexity until ultimately we get a grandmother cell. Okay? Now, it's true and not true. But the first step of it, they admitted, well, the second step of it, they admitted was not true. They said it goes from simple to complex to hypercomplex. And what is hypercomplex? Hypercomplex is going to be something which, what is it between simple and complex? Simple has to have this orientation exactly here. Complex can have it anywhere within here. And the hypercomplex says, oh yes, but only it has to stop here. If it's too long, then it doesn't respond. Okay? And then they found that there are also simple cells which have too long, it doesn't respond. So they called it end stopping. In other words, besides having an excitatory region in the receptor field of the simple cell, there can be an inhibitory region at the ends. And those inhibitory regions also are orientation tuned. So they only inhibit when there's line of that orientation there. So what's the response to this going to be? Big. What's the response to this going to be? Little. Now what happens if we have a curved line? This they didn't say. Somebody else found. What happens if we have a curved line? This one will not respond as well, and therefore there's less inhibition, and therefore we have something in between. And therefore a end-stopped cell is a good curvature detector. The more curved, the more it responds. Yeah?
The reason for that the middle part of the curve is more straight than the other part?
Yes, the middle is straight, and therefore there's a response to it, and these are angled, and therefore no response to it. Yeah, exactly. Okay? Say it again. Yes, if it was a parabola rather than a circle, maybe it would respond more. Okay? So. Okay, we'll do it later. It's okay, we'll take care of it later. It's okay, leave it. We'll do it after in the intermission. So a curved thing like this it will not respond to. A curved thing like this it might respond to. And a straight line will respond even better. Okay? Good. So it's a curvature detector, which might be very useful. There's some attempt to say that if it's end-stopped only on one side, then you might get responses to an edge of this kind. Yeah, maybe. This is part of the attempt of Yuval Weasel to answer everything. And this is from the book, so I put it in, but who knows. Okay. Next question. Can we detect within that or this? Can you see something in here? Yes, a row of lines. Up there you can't see it. It's supposed to be with zigzag. And here it's zigzag somewhere in here. I don't know. What's the difference? The point is that if there is a row, if cells with the same orientation are exciting each other, then we can see it. Okay? This is one kind of processing that might take place in order to allow us to see the continuity of this kind of a thing. Okay? Now this is very important. Why? Because if the zebra is standing behind a tree, you still want to see the zebra and know that it's one zebra, even though there's a blockage in the middle. Okay? So we want to see continuous lines, even though they're blocked in the middle. Okay? So this is not just an anecdote, but it's real. Okay? We have to be able to see things which are somehow blocked. Okay? You know that this is my arm and that the hand belongs to this arm, even though it's blocked here. Because you know that this line is continued. Okay?
They're exciting each other, you think?
They're probably exciting each other, yeah.
Or their input is something more high level than...
Here, this model says they're exciting each other, which is a very good model. There's work done by Gilbert, who worked with Weasel, who found that there are long connections which perhaps are doing this. Okay? Now remember, in order to do this, how long do they have to be? They have to jump from hypercolumn to hypercolumn, and they have to jump over all the columns of different orientations. Is that clear? Okay. This is one orientation column. This is a neighboring orientation column of the same, so it's jumping further away. So these connections have to be very, very selective. Okay. So in general, V1 has a very, very tough task in wiring. Okay. And the first thing that it has to do is align the two eyes. Okay. Now they're aligned already in the LGN, but still the inputs are separate and they come into two places, and then they want to combine. Why do they want to combine from the two eyes? Stereopsis. And for stereopsis, they want to combine with disparity, different levels of disparity. And the second is to strengthen, to know that we're seeing the same thing. They want to strengthen when we see things with two eyes rather than with one. Okay? So two reasons for strengthening it. Okay? So that's the alignment that it's making in that direction. But it also, once it's set up these orientation columns, it has to set up connections which jump over them. Okay? Difficult job. We spoke about that perception stereopsis, and you just mentioned it, and I just wanted to say here that the kinds of cells that are found are very often near or far cells. That is, stereopsis seeing anything that's closer than the point we're looking at, anything that's further than the point we're looking at, rather than necessarily tuned near and far. You would expect to have many different tunings. You don't really need it. You need just this and that. And then we can move and compare. I'm saying that the most important thing to know is whether something is closer to me or further from me than the point I'm looking at. Further, the z-axis. Okay? To the right or the left, that's done by the map. But closer or further is this. I'm looking at this point. If I'm looking at this, are you people further away than me than this is? That's what's important, so I know the relative depth. It's expressed here by having cells which respond only when it's further away or only when it's nearer. There are three types of cells. One which is narrowly tuned for further or nearer, one which is very broadly tuned for narrower or further. Okay? Okay. Motion perception. We mentioned motion perception last time, right? And we just said there are two ways of judging motion. One is by having the two signals with a delay summing, and the others with the two signals with a delay subtracting. The coincidence detector or the anti-coincidence detector. And we said that both exist. And we want to expand today a little bit about motion perception. The first point I want to make is, can this cell know the difference between something which is moving gradually or something which jumps from here to here? No difference. There's one detector here, one detector here. If it jumps from here to here, or if it moves slowly across, makes no difference. Okay? Jumping from here to here is what we call apparent motion. Okay? Now, apparent motion is what you see in Times Square, right? You have these letters moving across. Times Square? You've never been to Times Square? Or any animated film, or in fact, any film. Any film that you see is really apparent motion, right? You have a picture and then another picture, okay? And in Times Square, you have letters on this neon sign, which are just a lot of bulbs. Today, it's probably not, but anyway, it used to be a lot of bulbs. The bulbs turn off, and they turn on, and the letters are moved to the next position. You see it sometimes in neon signs. You have an arrow, and then an arrow, and then an arrow, and it looks like the arrow is moving. So that's called apparent motion. A few rules about apparent motion. If we turn off this spot and then turn on this spot, it looks like it moves from here to here. Okay? What happens if we have a circle here and a triangle here? Will we see the circle moving to a triangle? And the answer is yes. And where did it change shape? Somewhere along the way. So it looks like a circle which is slowly morphing into a triangle. Okay? What happens if we have a circle that we turn off and then we turn on two circles? It would look like it split and went into two places, or split to two places. What if we have two circles and then one circle? It'll look like they're combined. So apparent motion is very, very strong. Okay? It's one of the things you got to teach children is that when a person disappears and then reappears, it's the same person. Apparent motion does that for us. In fact, there's a strong tendency to see things as moving rather than as this disappearing and this appearing. Now, if you do it too slowly, if you turn this off and then turn this on, then it will disappear and turn on. Okay? Or if it's too far apart, this will turn off and this will turn on. So there's a limiting distance and a limiting time for seeing apparent motion. Okay? Good. Okay. Now, what about more complex motions. Okay. We don't need this. What about eye movements? Okay. So here's a spot, and it's moving along, and I'm following it with my eyes. Any motion on the retina? None at all. How do I know it's moving? Because I moved my eyes. So we have to compare eye motion with object motion. Or not object motion. Eye motion with image motion. So if the image motion is steady, but the eyes moved, then I know something moved. Okay? How do I know that my eyes moved? Very good. The motor cortex made them move. Okay? So therefore, the brain should know. Okay? The other possibility is that if I move my eyes, the whole world moves in the other direction. So I'm following this finger, and I move my eyes with it, but the world is moving across the retina. So there are two ways I could know. How can we differentiate between these two ways? Why do I need to differentiate? Because I want to know what the mechanism is. There are two mechanisms here. Either one is good. How do I know which one is correct or not correct? Yes. There are two mechanisms by which I can know that when I follow this, it's actually moving. One is that I told my eyes to move, and therefore I know that my eyes are moving, and the image is not moving, therefore it must be moving. The alternative is that the picture of the world in the background is moving, while this is not. So therefore, it's not the world that's moving, but this is moving. Okay? So how do I know the difference between these two? Do it in a uniform room. Okay, good. Go ahead. No background. Okay, yeah, that's one possibility. As a researcher, yes. You can move your eyes with your hands. You take your hand, and don't do this because it's very dangerous, and you move your eye like this. Okay? And if you do that, then you see the world moving. That's because you did not give the signal to move the eyes. The finger did. And therefore, when the eye moved, you do not correct for it. Therefore, it must be that you're using what's called the collateral signal, the signal that comes from the motor cortex that tells the eye to move. It's telling also the visual system, interpret this not as a moving world, but as an eye moving. Okay? There's another way. Paralyze the eyes. Okay? If you paralyze the eyes, then when I give a signal to the eyes to move, will the eyes move? No. And therefore, what will happen to an object I'm looking at? It'll appear as if it's moving in the other direction. Okay? Now, this is a very dangerous experiment to do, because the only way of doing it is to paralyze the person. If you paralyze the person, he can't breathe. If he can't breathe, you have to give him artificial respiration. So therefore, this is an experiment which is only done by vision researchers on themselves. Okay? And what does it feel like? Well, if the respiration is a little bit too fast or a little bit too slow, so you tell the guy who's helping you, make it slower or faster, right? You can't tell him because you can't speak. You're paralyzed, okay? So it's really a horrible feeling, okay? So, therefore, it's only done.
But the moving world is also a mechanism, isn't it?
The moving world is also a mechanism, but this one is much, much faster. How do I know it's faster? We've done this experiment already. Move your hands like this, and you see a very blurred finger. Move your head like this. It's not blurred. Why? Move at the same speed as I move my head. This one is not blurred. This one is blurred. Why? We don't follow with the eyes. Why not? Not as fast. Why not? Why can we move the eyes very fast when we do this? Vestibular system. The vestibular system has the three semicircular canals, which are linked to the three directions of eye movements, and they are very, very fast to move the eyes back. Okay. Next question. What about complex motion? I'm going to move this to the right. Now, every receptive field only sees one little portion of the world. So it only sees this line. And as this moves this way, this line moves across the field. So it looks like it's moving in this direction. If I'm moving that way, because within it I only see the line. The line can't move this way, right? It can only move that way. Got it? No. Yeah. So you can see this blue thing inside here.